How's everybody doing?
Next.
Is it just me, or does it feel like we're devolving into complete and utter chaos? Complete and utter chaos. Awesome. Great. I'm glad I'm not the only one. All right, it looks like a number of you guys have gotten your VNet-OWL summaries turned in. If you haven't, get that done here pretty soon. Where's Dr. Judd? I don't see Dr. Judd. Anyway, if you look in your syllabus, in the section of the syllabus that deals with literature summaries, it says in there that the first one is going to be graded by Dr. Judd and I. The second one, you're going to do a peer review of one another's papers. So expect later on today, after I've downloaded all of your literature summaries and changed the names on those of which don't have the right names on the file and things like that, I will send you out a literature summary from another student in the class. And what we want you to do is we want you to go through there and use two resources that we have on Moodle, on the Moodle site. One is the rubric that we would normally use to grade your grade your literature summaries. And the other one is that list of common writing mistakes that students make. And what we want you to do is go through and evaluate the literature review that you receive and get that back to us by next Friday. So you have a test coming up on Monday, so we don't want you to spend your time on the weekend doing that, but we'll give you a week to work on that. And then your final grade on this literature summary will be comprised, half of it will be the results of kind of how your literature summary was. So it'll be our evaluation of your literature summary. And then the other half of your grade will be based on how thorough and thoughtful and constructive your review of someone else's literature summary is. The reason we have you do peer reviews is because seeing other people's writing helps you get ideas about your own writing. And also being able to kind of look at somebody else's writing through a critical lens trains you a little bit in how you can turn that same eye on your own writing. And so you'll do peer reviews periodically over the course of your career at Jewel. You'll do a couple of them here in BIO133. Nathan, you look like you were about to say something.
I was going to say, since you have a week, don't forget to use that other required textbook, The Short Guide to Writing in Biology. Chapter 6 can be a really useful resource for giving thoughtful, critical feedback.
Whatever you paid for that book, chapter 6 is worth the cost of the book.
The other thing I was going to add is that you should all receive responses from me or graded versions from me of your C3, C4 homework later this afternoon. So you'll have that, have those feedback to help you study for the exam.
NOVA homework should also be coming to you very soon as well. I've got 4 more of those to get through, and then I'll send those out as well. There's a key to the ANOVA homework on Moodle. It's on the day that the homework was assigned. So if you scroll up to where the homework assignment was there, there's a very thorough key walking you through those calculations and things like that. Clearly, you're not going to have time on an exam to work through an analysis of variance, but I want you to understand how they operate. In particular, I want you to be able to interpret an analysis of variance table, which you should have constructed as part of that homework. And then the other thing that I want you to be able to do is I want you to be able to interpret graphs that have been that illustrate data that have been analyzed with analysis of variance, so that you can interpret whether things are different or not. And we're going to do some graph interpretation today as part of the Veen et al. Discussion. Nathan.
Yes.
I'm going to make you host so that you can set up the breakout groups while I'm finishing up the last little bit of nitrogen cycling. Is that all right?
Okay. Yeah, let me make sure I have them still. I usually have them printed out and I'm at home today. So let me make sure I have this in my email here.
All right. Any questions from last time? I feel like we blew through the nitrogen cycle at light speed. Because like, yeah, you did exactly that. All right. I'm going to back up just a little bit today to get started, but then we'll be on. Assuming that I can find where it is. I've made a couple of changes to this PowerPoint, and I'll upload those to Moodle after we're done today. I just didn't get around to doing it this morning. I had another catastrophic day of problems with an exam in my CTI class, so that kept me busy long past when the test was over. So.
1, 2, 3.
Why does it always do this? Okay, can you guys see? No, you can't see. Can you guys see a very gnarly looking nitrogen cycle on your screen?
Yes.
Yeah. Okay. All right, cool. Gnarly is good. So all of those fluxes and things that we went through were basically to get us to the point where we can look at a cycle. And so, for example, we have here atmosphere in the form of N2. There's a ton of it up there. There's actually gigatons of it up there. And all of this atmosphere gets into the terrestrial environment through nitrogen fixation, whether that's through plant roots or whether it's by free living bacteria. Once it's in plant roots, it can be used by plants. It can be added back to the soil through death and decomposition. It can also be, plants can be eaten by animals. So this is another one of those arrows that this particular model doesn't have simply because I neglected to put it in there. But consumption by animals will also move nitrogen from plants into the animal pool. Animals as well as fungi excrete nitrogen-containing compounds into the soil, and then those things eventually get broken down into things like ammonia. We have dead organic matter that is coming from the death and dying bodies of these animals. And that dead organic matter through decomposition gets converted into dissolved organic nitrogen. Then that dissolved organic nitrogen gets converted to ammonium. And that then through nitrification gets converted into nitrate. Nitrate and ammonium and all these things that are capable of being attracted to water because water is a polar molecule then have a possibility of being leached. This ammonium is going to be blown back off into the atmosphere through the process of of ammonia volatilization. NO3 gets blown back to the atmosphere in the form of N2 through denitrification. And then of course, there's also volatilization by fire. So it's a very complicated cycle because nitrogen can take on multiple forms and nitrogen goes through multiple transformations. Now, many of these transformations are in soil and they're accomplished by different kinds of bacteria. And so in that way, a lot of what's going on in the nitrogen cycle is actually all happening down here in soil. And there are only a couple of interactions between the terrestrial ecosystem in this case, and the atmosphere, which is the major pool. So I just want to set your minds at ease in a couple of ways. One, we're not going to ask you to draw this from scratch on the exam. That would be ridiculous. We're also not going to ask you to draw the carbon cycle on the exam from scratch. What we will do, though, is we will give you, in some cases, the players and maybe what some of the flows are and things like this, and ask you to label which fluxes are which fluxes. So we would have, for example, this same diagram, and instead of having the names for all the fluxes, we might have boxes there where you can put in what the fluxes are, and we would generally give you what all the fluxes are that you could choose from, recognizing that in some cases a flux may appear more than once on a particular diagram. That's especially true with the carbon cycling diagram. With the carbon cycle, because we concentrated on how that carbon cycle either stores carbon in the absence of humans or exports carbon back to the atmosphere in the presence of humans, we might make you do some math. But once again, we wouldn't make you recall what the values of all those fluxes are. We would give those fluxes to you and then expect you to just be able to use them to demonstrate to us why an ecosystem is storing carbon versus exporting carbon back to the atmosphere. Another way of kind of wrapping your head around this information is on the next slide, where I've basically just identified what all of the pools are and what all of the fluxes are. So we have an atmospheric pool, we have plants, we have animals, we have microbes, we have dead organic matter, we have the soil, which is a huge and complex pool of nitrogen for this particular nitrogen cycle. And then we have groundwater that's basically going to flow out into stream flow. And then we have all of these fluxes. A good thing for you to do would be to go through this list and identify which of these fluxes actually involves a transformation and which of these fluxes are just the movement of nitrogen in one form from one part of the ecosystem to another. So in some cases, these fluxes, like nitrogen fixation, involve a transformation of nitrogen, but in other cases, things like excretion don't really involve the change in form of nitrogen, nor does death for that matter. So another way of thinking about this is something that I told you should try doing over the time between Wednesday and now, which is to basically just put these things into a table. And so what I've done here is I've created a table for your reference. It basically has the flux, nitrogen fixation. It begins with N2 in the atmosphere, and the N form of this flux is ammonia in the soil or in the roots of plants. If you're looking at these leguminous plants that are that have a close association with nitrogen fixing bacteria. Who accomplishes this transformation? Well, it's actually the bacteria in those roots, or it's bacteria that are free living in the soil. And this transformation occurs in the soil and root nodules of the legumes, which is where these bacteria live. Similar thing for ammonification. What do you start off with ammonification? You start off with dissolved organic nitrogen in the soil. That gets converted into ammonium. Once again, this is done by a group of bacteria. Now, these bacteria are different from these bacteria over here. And so keep in mind that these are different taxa of bacteria doing this. And once again, this happens in the soil. Where you might ask yourself, well, where does dissolved organic nitrogen come from? Well, that comes from decomposition down here, where when organisms die, the organic matter in their dead bodies gets broken down by decomposers and dissolved organic nitrogen is released. This is done by bacteria and fungi. I should have put fungi in here, and invertebrates. And once again, this happens primarily in the soil. If you look in the notes on this particular slide, there's a description as to why these things are colored different colors. So all of the fluxes that are in blue involve either fluxes that involve either bringing nitrogen into the terrestrial ecosystem from the atmosphere, or they're all fluxes that involve what we call intrasystem cycling around inside the terrestrial ecosystem. And the two that are in black, denitrification and volatilization, those are the two fluxes that are involved in taking nitrogen out of the terrestrial ecosystem and putting it back into the atmosphere. And so this is a way that you can go about systematizing the nitrogen cycle in a way that might make it easier to remember what is by far the most complicated cycle of all of the biogeochemical cycles. So I've basically broken it down into what's the flux, what's the beginning form, what's the end form, who does it, and where does this happen. And those will be similar for all sorts of sorry, those will be different for each different flux. Assimilation involves no transformation. You're just absorbing the nitrogen form that is in the soil. This is done by plant roots, and that goes on in the soil. You'll also see that for excretion can happen in a variety of different forms, but because those forms are all there in the beginning form and the end form, once again, there's no transformation in that flux. It's just a movement, in this case, from organisms into, once again, the soil. One of the other things that this way of looking at the nitrogen cycle should demonstrate to you is what's a pool of this resource that is actually super important in terms of understanding the nitrogen cycle. What's probably the most important pool to think of? Nobody? Should be really apparent from this table.
The soil?
Yeah, soil is a super important place. So when you go back and look at this diagram, this box of soil it's large because there's a lot of nitrogen in there, but it's also large because I had to make the box large because there's a lot of movement of nitrogen going on in soil. There's a lot of changes in nitrogen, changing from one form of nitrogen to another form of nitrogen that is going on in the soil. So sometimes tables can make things like that also jump out to you. Nathan, how important do you think human perturbations to the nitrogen cycle are? Can't hear you.
Yeah, I suspect they're very important. We like.
All right,
so
then I'll run through a couple of examples. So just as humans perturb the carbon cycle and take it from a cycle that tends to store carbon in soils and instead releases carbon back to the atmosphere. So there's a net movement of carbon into the atmosphere. In the same way, humans have been altering the nitrogen cycle for hundreds of years, actually, as it turns out. A lot of this is done through agricultural activities. And so these are a couple of papers that are based on some long-term plots in Illinois. And these plots are plots where they started adding nitrogen to the soil in the same way that farmers would be adding nitrogen to the soil. And one of these papers deals with the effect of that on carbon in the soil, and the other one deals actually with nitrogen in the soil. And the reason that I want to highlight these two papers is that these two papers kind of lead to some counterintuitive sorts of conclusions. And so right here you can see the organic carbon that is in the surface 15 centimeters of the soil. And this represents the point at which they began these fertilization experiments. And what you see generally is that in these fertilization experiments, you see that there is a decline in soil carbon as you add nitrogen to soils. And so one of the things that you might ask yourself is, well, why does adding nitrogen reduce the amount of soil organic matter that contains carbon in the soil? When you add nitrogen to soils, what's one group of the soil community that you're going to basically be feeding and enriching? Once again, there are probably lots of clues in this paper.
Bacteria?
Yeah, you're going to be feeding bacteria, and many of these bacteria that are in soil are involved in breaking down organic matter. They're decomposers. So if you're enriching the soil with nitrogen, you're feeding these microbial communities. The amount of cellular respiration that these microbial communities are doing goes way up, and they tend to break down all of the organic matter that is in the soil, which basically reduces the amount of soil carbon that is found in these ecosystems. And so we can have these situations where what's happening in one biogeochemical nutrient cycle can actually affect what is happening in another chemical cycle. And this, of course, is one of the reasons that we should think about nitrogen when we think about carbon exports to the atmosphere, because as these bacteria get fed by nitrogen, they're breaking down more carbon, they're returning that carbon to the atmosphere more quickly than they would if we weren't fertilizing these fields. But what's really kind of freaky deaky about this is that when you look at nitrogen, turns out that the more you fertilize your fields with nitrogen, the less nitrogen those fields have. And once again, one would ask, why is putting nitrogen on your fields actually depleting those fields of nitrogen? And there's a couple of reasons. One of the reasons is that the form of nitrogen that you put on your fields is being put on there because it's easily assimilated by plants. But when it's easily assimilated by plants, it's also prone to leaching. And so a certain amount of that nitrogen just leaches right out. Whatever doesn't get picked up by the corn plants that you're applying it to gets leached out. But once again, let's go back to Dominic's idea. What happens when you add nitrogen to soil, to the bacterial community?
They break it down.
Yeah, they're going to absorb it and they're going to use it for energy. And so these denitrifying bacteria, in denitrification here, they're going to basically be taking that ammonium nitrate, and that's generally the form that we have nitrogen in commercial fertilizers, and they're going to break down that nitrate and they're going to convert it back into atmospheric nitrogen and blow it out of the soil through the process of denitrification. And basically, the more you add nitrogen fertilizers to your fields, the more you build up communities of denitrifying bacteria, which means that they break down the nitrogen that you're actually adding to the soil, but they also break down any nitrogen that is going into the soil from just normal processes of the corn plant gets harvested, but then all of the corn residue gets plowed in. That corn residue has a lot of carbon and a lot of nitrogen packed into it, the bodies of those dead corn plants, but they're going into soil that has this supercharged bacterial community that then breaks that stuff down very quickly and releases all of that nitrogen back to the atmosphere. And so what they found over time is that, strangely enough and counterintuitively enough, that the long-term result of putting synthetic fertilizers on agricultural fields is that the amount of soil nitrogen actually goes down over time. And so that is kind of freaky weird, but that's the way it is. We also put a lot of nitrogen in the atmosphere through the process of burning fossil fuels, and that also can have a fertilizing effect on ecosystems as well. Yeah. Nathan, you were going to say something?
I'm going to add something in here. So this is a really good discussion of how nitrogen fertilization can perturb ecosystems in surprising ways at the small scale, on the scale of an agricultural field. Remember that most of North America's prairies have been converted to agriculture. So there's a lot of nitrogen being added all the time. One of the large scale effects of adding all this nitrogen and much of it leaching out into the waterways is that at very large scales, when you get nitrogen fertilization of aquatic ecosystems, like at the mouth of the Mississippi River or the mouth of other major rivers around the world, you get another interaction between a biogeochemical cycle. All these organisms that are using nitrogen and using carbon are also taking up oxygen. So you get these vast areas in the Gulf of Mexico, in the North Sea near Europe, and in off the coast of China, off some of those major rivers, and in India, where the water becomes anoxic because all the microbes use up the oxygen while they're using up the nitrogen fertilization. And that can seriously disrupt ecosystems and food chains in marine ecosystems and cause what we call dead zones, where there's very low productivity, very little fish, not a whole lot of good productivity there for people who live and depend on the coast.
Yeah, if you search for Gulf of Mexico dead zone, there's a lot of news coverage about the Gulf of Mexico dead zone that is due to the Mississippi River dumping into the Gulf of Mexico and carrying both nitrogen and phosphorus nutrients into the Gulf. So this last figure here, this last picture, is a picture that I took in the Wichita Mountains in Oklahoma. And you can see that this is a prairie. And this actually happens to be a prairie that is the home of a very, very large prairie dog town. And one of the things that prairie dogs do is they dig tunnels and burrows. And in the process of this, they've brought a lot of the deep soil to the surface as they've created their prairie dog town. And one of the things that happens is you get a different plant community growing on the landscape where this prairie dog town has basically brought nutrients up from the deep layers of the soil so that they're now near the surface of the soil. And so this area of plants that seems to be doing really well in here, that group of plants almost perfectly covers the geographic area that is impacted by these prairie dogs, because they've changed the nutrient availability at the surface of the soil such that these plants can compete well there, because these are probably high nitrogen requiring plants, because they've gone down and they've brought up those deep layers of soil where all of that nitrogen has leached down to the bottom. And as a result, you get this very distinct shift in plant community structure as you move from the prairie without prairie dogs to the prairie where the prairie dogs have dug their town. And it's just something that I noticed while I was there once, and it just made for a nice photo. And that is that. Any questions? All right. On the Moodle site, there's something in the chat, which I cannot see until just now.
Yeah, I figured you'd see it with the questions. I've got about half people assigned, and I'm stuck. So
okay.
Let's have people go to the Moodle and find the questions that they're going to discuss. Is that right?
Yep. Go to Moodle, find the questions. They're posted on today's date, and we'll get you put into your groups.
I'm going to open the rooms. Some of you are going to get notification, and then a few of you are going to be left behind, and we will add you in manually while you're doing the Moodle lookup.
Yeah, let me pull up my list. I don't think I have a hard copy open right now.
I only have it on paper, and it's in my other folder today.
Okay. I actually have it. Oh, but you need it, Nathan, don't you? Because I can't.
Yeah.
I can't do it.
So I have about. Can I just read people off?
Yeah. Yeah. Sure.
Okay. Riley Bant.
Room 2.
Casey Behrens.
Room 3.
Tavares Albury.
Room 1.
Audrey.
Room 1.
Trying to go mostly alphabetical here. Brandon Oman.
Room 1.
All right. Michaela Massey.
Room 6.
Almost there. Keaton Porth.
Room 2.
Devin Sparks.
Room 6.
All right. Dominic Lee.
Room 3.
Cody Oshiro.
Oh, I just saw him. Room 1.
Last two. Ryan Woodson.
And Connor are both in
4.
room 1.
Oh. In room 1, okay. Oh, not everybody's here today, I guess.
Wait. No. Sorry.
Oh.
What's going on? Oh, why do I have
Connor.
that up there? Sorry. Connor and Ryan are both in room 4. Sorry, room 4.
Okay, that makes sense.
All right.
All right, that's good. Everyone's there.
And stick me in a room, and I'll let you know when I need out.
Yeah, okay. Yeah. Let's do. Right.
Yeah, so yeah, I'm in quarantine now, so
Sorry.
no, it's no biggie. All right.
I feel like we're missing one more.
Alex isn't gonna be here.
Alex is never going to be here.
Yeah.
Did he drop out?
Yes.
He did drop. Yep, he dropped the class.
Okay, you guys ready for the questions?
I guess so.
Yeah,
You guys all have them pulled up, or do I need to read them?
I haven't pulled up.
I have him right here with me.
Awesome. Okay, so effects of fire, I guess we could start with.
Decrease diversity.
Mm-hmm.
Well, in short term, they
I need to pull up my paper because I can never remember which one's which.
so for the plots that got like the 1-year burns, the diversity was much lower than the plots that had 4-year burns, which makes sense because the 4-year burns had more time for the slower spreading plants to kind of recover versus
Yeah.
Also think about in those low diversity areas, what were the main kinds of plants that ended up dominating those 1-year burn return frequency plots?
Safe
Grasses, like the 5 before grasses that they listed in the introduction, or
Yeah,
method.
it ends up being dominated by grasses. So you can think about the structure of the plants and where the meristems are on the plants, and that will give you some clues as to why that happened. Where are meristems on grasses versus forbs? Remember, meristems are those parts of the plant that are growing, doing cell division to increase the length of the plant.
Okay.
I'm sorry, I didn't catch any of that. My internet is very bad at home.
Okay. If you think about if you think about where meristems are, the growing portions of the plant, that'll give you some clues as to why fire affects forbs more than it affects grasses.
Okay.
So where are the meristems of grasses and where are the meristems of forbs located?
Isn't grasses lower than forbs?
Yeah, grasses are located down at the ground level, and forbs are located out at the tips of the stems. And
Yeah.
so when a fire goes through an environment, it leaves the meristems of grasses intact and it destroys the meristems of forbs. And so grasses are going to respond to fire more easily or recover from fire more easily than forbs are. And so that's why you get these low diversity communities dominated by grasses. It's just a matter of kind of how the plants themselves are structured.
Yeah. Okay, do we want to move on to species diversity because of grazing?
Sure.
Okay. That one, I'm pretty sure.
So in the areas that there was grazing had much better diversity than areas
Yeah.
that didn't. There's much more dominance in areas without the grazing.
Okay, for number 6, what measurements were made in each of the 0.25 meter squared plots?
Well, we know they measured the light availability as well as some of the prairie animals and their urination and dung piles that were in there. And then the nitrogen in the soil as well.
And then the second part to that is, how is community structure in each plot assessed?
And part of that, it says that they measured plant species composition and they estimated it visually. It says that on the beginning of the measurements section.
So number 7 is how they basically.
But wait, that doesn't get at heterogeneity, which I think was the question that you asked, wasn't it, Casey?
Mhm.
Yeah, it was the how did they assess community heterogeneity.
So what do you mean by heterogeneity, and how would that be assessed?
Basically, like the diversities of each species within the community.
Well, diversity is different than heterogeneity.
Isn't it how like, okay.
Go ahead.
Isn't like an aspect of heterogeneity how like dissimilar each of it is though?
It is. Let me get back to the Zoom screen and let me share something with you. something that I made for another student because another student asked about this, and I needed to have a visual to kind of illustrate it. So can you guys see my PowerPoint?
Yeah.
So imagine that these are species richness values for a bunch of little plots, a bunch of little maybe quarter-meter square plots. In this plot over here, that's 12, 13, 15, 11, 12, 13, 14, 14, 12, 10, 11, 15. But over here, it's 12 species, 3 species, 25 species, 11 species, 12, 22, 23, 5, 7, 4, 3, 8, 1. Which of these two plots is more heterogeneous?
Is it the one on the right?
Yeah, it's the one on the right, because those plots are all dissimilar from one another in terms of the number of species that they have in them. So just with something as simple as a count of the number of species, it may be that these 12 species are different from these 13 species, but these have 12 species, these have 13, these have 11. Those things are not that different from the standpoint of species richness. And so similar thing if you did it with species diversity or whatever else. So imagine that this is one of those plots and each of these is a 0.25 meter squared plot. You can look within that plot and calculate how dissimilar those little plots are to get some measure of how much heterogeneity there is. The more dissimilar they are, the more heterogeneous they are.
So how large of an increment does it have to be classified as heterogeneous?
Well, it's on a continuous scale, and you can actually calculate it by, there are a number of dissimilarity indices out there that allow you to numerically compare how similar or how dissimilar two data sets are from one another. And quite frankly, I don't remember which index that they used.
So then to answer the question, it would be more, they looked at the percent cover of each plant and then compared that to the rest of the plots and said, this has more variety in this types of species than this plot does. So it's more heterogeneous or?
Yeah, so in each of these plots are rectangular. And then within these plots, they have a bunch of these little subplots, these 0.25 meter squared subplots. And what they basically said was, how different is this one from that one, 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 that one, that one, that, and that, for all possible combinations of these. And then they generate a dissimilarity index for this plot, these subplots within that plot. So that's why this question appears when it's talking about the little 0.25 meter square plots. They use those to look at how much does this little quarter meter square differ from this other quarter meter square? How do those differ from this other quarter meter square? And that's why they refer to this as small scale heterogeneity, because this is all done at pretty small scales. Quarter meter square is just not that big. Does that help?
Yeah, that helps. So number 7 talks about how they measured ammonium and nitrate availability, but only reported the results for nitrate. And so based on your knowledge of the nitrogen cycle, do you think this is okay? Why are we not? I feel like they didn't report it because the beginning form of the nitrogen, or the beginning form of nitrogen is gas since it's in the atmosphere, and ammonium is like just already found in the soil.
But it's not asking about N2, it's asking about nitrate. So NH3 and nitrate are both found in, so ammonium and nitrate are both found in the soil. Why did they concentrate on nitrate? And they don't ever tell you why they concentrate
Is
on
it
nitrate.
because ammonium is in the urine?
So
Well, so certainly it's in urine, so it might actually be more prone to being like locally abundant, depending on where bison and things like that have been. So that
Right.
might be one reason. Sure. What else?
in the paper, because I picked it up to check, it says that the resin collected in the nitrate, but it didn't collect the ammonium.
Okay, so the way that they collected nitrogen, they had to do it in a way that they weren't disturbing the soil, because to disturb the soil would have disturbed the plant community, et cetera. And so these resin bags only collect nitrate, and so that's the only thing that he could measure without going and totally disrupting all of the other aspects of their experiment.
So I think that would have been where the plots were.
Yeah, I think you're right. So how
Okay.
was each structure in the plot assessed?
Didn't they have like grids within the plots?
What do you mean by grids?
Like little, well, it talked about like 2x3, like so that it could measure. You know?
Yeah.
Yeah, that's what I meant.
I mean, I think I remember something like that. I just, don't.
Something like that.
Yeah.
Aren't you glad I dropped in? And they talk about this in the setup. Continue.
So is that grid referring to like where they measured everything, like the exclosures and the other like sample size, like 25 meters or something away?
Yep. So this is have a watershed. At the top of the watershed, you'll have exclosures. And then if you blew that exclosure up, there's a measurement area inside that exclosure, and inside that measurement area is a bunch of subplots. And then outside the exclosure is another measurement area that has the same number of same size subplots. And they measured everything that they measured inside each of these little subplots. And I forget how many subplots they had in each exclosure. Yeah, so they measured diversity, they measured nitrogen levels, they measured all this stuff within each of those little subplots. Yep.
So then didn't they assess like those different characteristics of the ecosystem? They did like the resin bag and they did the like light septometer, and then they did the test with the like compared the fire versus the
Yeah, so these are set up in both the 1 year burns and the 4 year burns. The exclosure keeps the bison out, so that creates the grazing versus non-grazing thing. But then within each of these plots, though, they take lots of little samples. So the next part of this question is about community heterogeneity. Why take a bunch of little samples in a small area rather than just sample that area once?
Well, if you do like multiple, I feel like little like samples, then you'll get a better like common answer, because if you just did it once, there could be other variables that played into that.
Okay, but the real reason relates to heterogeneity. How do they assess heterogeneity? What is heterogeneity?
Isn't like heterogeneity is like the amount of species and the like variance of species within like a group? So there can be like different groups of little micro communities within a larger community.
So if this is inside the exclosure and this is outside the exclosure, and there are 12 species in this plot, 13 species in that plot, 14 species in that plot, 11 species here, 10 species there, 12 species there, 15 species, 13 species, 14 species, 11 species. But then outside the plot, there's 2 species, 22 species, 17 species, 4 species, 9 species, 16 species, 24 species, 18 species, 3 species, 1 species. Which of those areas is more heterogeneous in terms of the number of species?
The one with the most amount of species.
Nope, that's just the most species rich. But these are all very similar to one another in terms of the number of species that they have. These are all very dissimilar to one another in the number of species that they have. This one is more heterogeneous than this one because these are all very similar. These are very different from one another. And so if you only measured this plot once, you couldn't measure heterogeneity because you would only have one value for species richness in there. Does that make sense? And this is also why they refer to it. So for those who were in the group that I was just in, I was telling them this is why it's small scale heterogeneity, because it's at a scale where they have a bunch of little plots in these bigger plots, and they're looking at heterogeneity from one subplot to another subplot. Rather than just sampling the entire plot, they divided it up so that they could look at this small scale heterogeneity. All right, Dr. Judd, you want to do the debrief, or you want me to?
I can get it started. Maybe we can bounce back and forth a little bit.
Okay.
So what do you guys think of this paper compared to your experience reading the Henry et al. Paper?
I think it was much harder to write about.
This one was more difficult to write about, you thought?
Yes.
Yeah.
Yeah.
Why is that, Keaton?
Do what?
Why did you find it harder to write about?
I don't know. Maybe it was just the subject matter wasn't as intriguing, I guess.
Okay.
Could be.
Somebody else was saying something?
Yeah.
Yeah, I agreed with him. I feel like the reason that it was difficult to write about was just like the understanding of like what the vocab words like specifically really meant. Like, even though you guys did go over that stuff, like knowing the difference between like diversity means this and this, or like evenness, and just like all of those different vocabulary words combined were just like, it was a lot, and it was hard to keep them straight when you were trying to write about it.
Riley, did I go through diversity versus heterogeneity in your group? Was that your group that I did that with?
No.
No, it must have been some other group. But yeah, like what's the difference between diversity and heterogeneity? Well, they're actually very different things. Until you get that straight in your head, it's a little difficult to deal with it. Yeah.
And how about, so there are a lot of things, a lot of moving parts in this paper, but what was the experience like reading it compared to reading the Henry et al. Paper?
I think reading is way easier for the second paper because, well, the PC one, I don't really remember, was really new for us. So I think it was the problem for the first paper. And also like the second time, we already had some experiences of reading a research paper.
Okay.
I agree. I think it was a lot easier to read, but I thought it was like harder to write about because it had a lot of like details that I felt like were important, and like obviously this paper is concise, and so I had trouble like getting it down into like concise words that made it clearer.
That's a real challenge, taking what is a paper that does have a lot of moving parts in it and boiling it down to its essence. You have to go through this process of deciding, well, what level of detail do I actually need to include as I write about it versus what things can I get away with leaving out or glossing over and things like that. And that's a skill. Imagine when you go to write your final research paper in here, you're going to have to, in some cases, summarize an entire paper in a sentence or two.
All right, and one of the things they focused on for this paper was interaction effects. Can someone explain what an interaction effect is? There's two key ideas that many of us have talked about are the fact that this study was aimed at the small scale, which Dr. Quincy was just talking about when we started this group discussion. And the other aspect was looking at interaction effects. So I want to make sure you guys understand that.
So this might be a totally awful explanation, but I think I remember like when you're looking at the graphs, if you like draw a line from the middle of each bar and they cross and there's an interaction between the two. Is that totally wrong?
We did use that, that sort of scheme to discuss what interactions are and how they work. But you can also think about it in terms of the bar charts that are in the VNet all paper, which another way to ask this is, which things did they measure, like diversity, coefficient of variation of nitrogen availability, and community heterogeneity? Of all those different bar charts, which ones showed interaction effects between grazing and burn frequency, and which ones didn't? And how do you know which are which? For example, they predicted, right? One of their hypotheses was that you'd have highest diversity at a 4-year in a grazed area. Was in fact diversity highest in the 4-year burn and grazed category treatment? No, Paul has it here.
The answer is found in figure 1B to Dr. Judd's question.
Yeah. Is diversity really higher as they expected? Is it really highest in the grazed, burned every 4-year treatment?
Isn't it insignificant, like regarding whether it was burned or not? The diversity is found in whether it's grazed or ungrazed.
Good.
Yeah, but is there still an interaction in that figure though, figure 1B?
So you answered the question correctly with respect to the hypothesis about looking at the grazed category, but now look at the ungrazed category. How is that different?
In the ungrazed, you can see the 4-year burn has a higher diversity than the annual.
Yeah, good. So that's the key to the interaction. Whether you can see the effect of burning is conditional on whether you're in grazed or ungrazed.
Yeah, that's what I was working on when I had that other figure on my board. If you're looking at my board, while we didn't see an interaction in the grazed because of the grazed, we still do see an interaction because of the ungrazed. And what that means is basically that the 1-year and 4-year burns are doing different things under grazed conditions than they do relative to one another they're doing different things in the ungrazed areas than they are doing relative to one another in the grazed areas. And that's the very definition of an interaction is the response conditioned on some other factor? So do these two things behave differently in one set of treatments versus how they behave relative to one another in another set of treatments? And another way of thinking about this is if you draw lines from the means, do those means, are those lines parallel or not? They don't have to cross, but if they're parallel, it means that the same thing is happening in both sets of treatments. If they're unparallel or crossing one another, either diverging or crossing, then the two treatments are doing different things.
So I have I wanted to talk briefly about the nutrient or resource portion of the paper, but is there anything else, Paul, before I go to that you want to review here?
Yeah, so the things that I would want to say about figure 1 in particular is that figure 1A, 1C, and 1D are all different aspects of what gets rolled into diversity. So diversity includes the number of species, which is species richness. Diversity includes evenness, which is how evenly distributed individuals are across those species categories. And dominance is just the opposite of evenness. Dominance is how unevenly they're distributed across those different species categories. But both species richness and species evenness are part of species diversity. And so within those figures, you should be seeing that as richness goes, so should diversity and so should evenness go. And as each of those are moving one direction, you should see dominance moving in the opposite direction. And so think of those figures as integrated with one another. And then the other thing that I would just say to you is don't forget about heterogeneity and how they quantified it. And what they found was that where grazers were present, they found more heterogeneous environments than they found where it was ungrazed. And once again, what I've been stressing to people as I've been talking about it in class is think about the aspects of the plants that were involved in that heterogeneity. In the low diversity plots, what kinds of plants dominated those low diversity plots?
C4 grasses.
Yeah, these C4 grasses. So grasses have meristems that are located such that they're not damaged by either grazing or fire. And so when you have fire running through a place, you end up with less diversity because those meristems are protected, and so only grasses survive. And grazing, especially if it's done by a selective grazer that likes grasses more than it likes other things, the grazing aspect then allows those non-grass species to get a chance to survive and succeed. And so as you're thinking about these graphs, don't just think about them as data, think about them as being caused by the organisms that are actually kind of embedded in the data. Think about the biology of the organisms and why that answer turned out that way. Go ahead, Nathan.
The last thing I'll say when it comes to the resources portion is I want you guys to think about the four hypotheses, the four specific hypotheses. Ask yourselves whether their data supported or did not support those hypotheses. And then once you've done that, go back and ask yourself, are you surprised that their data did or did not support the hypotheses? And one of the places where you can really get a fruitful sort of fodder for maybe critical thinking is looking at the pattern that they found in resource variability. Was that affected by grazing or fire or an interaction effect? And if so, what did that look like? And if not, are you surprised that it wasn't the case? And what might you do? What might you have done differently if you wanted to detect a signal that really is there? But we're right out of time, so I wanted to leave you guys with that little inspiration of something to look for. And we will, yeah, go ahead. We'll get back together on Monday.
There was a question from an email about a review sheet for the exam. I haven't started on it yet because I was dealing with CTI 259 exam, but I'll start working on that today. I'll ship what I have to Dr. Judd, and we'll try and have something out to you relatively quickly.
That reminds me that I was worried about the timing of both putting one of those together and also responding to an email that we got from some students asking about a review session this evening or something like that. Yep. I'm available this evening, but we also have to work on the review sheet. So
yeah, well, I'm going to get started on it as soon as I get some food in me. So
and maybe we'll email back and forth with each other and send an announcement about a review session if we want to do something like that for them.
Sure. I think the only a couple of people have contacted about us, and the main thing was make it after 4:30 or something like that.
Oh, okay. Yeah.
So
I can do any time. So we'll send an email out a little bit.
Okay. All right. All right. We'll see you guys on Monday. Let us know if you have any questions between now and then.